Hello students, hope all of you are fine. So dear children, we are doing the lesson biomolecule. Okay, so under this we had discussed the monosaccharides. Okay, and today we are going to discuss the polysaccharides. Polysaccharides, they are complex carbohydrates which are formed by polymerization of large number of monosaccharides, monomers more than 10 but usually numerous. Polysaccharides are also called glycans. They are long chained. The right end of polysaccharide is reducing end, while the left end is named the non reducing end. The chain may be branched or unbranched depending upon the composition. Polysaccharides are of two types, homopolysaccharides and heteropolysaccharides. Now, what are homopolysaccharides? Homo or homoglycans, okay, homopolysaccharide or homoglycans are those complex carbohydrates which are formed by polymerization of only one type of monosaccharide monomers. For example, starch, glycogen, and cellulose. They are composed of single type of monosaccharides called the glucose. Depending upon monosaccharide unit involved, the polysaccharide is called glucan, okay, glucan made of glucose, fructane that is made of fructose, xylene having xylose, arabin made up of the arabinose, galactone formed by galactose. Okay, we can see the example monosaccharide and disaccharide. Now, heteropolysaccharides or heteroglycans are those complex carbohydrates which are produced by condensation of two or more type of monosaccharides or their derivatives. For example, agar, chitin, peptidoglycan, arabenogalactans, arabenoxylans, etc. During the formation of polysaccharide, a molecule of water is released at each condensation. Okay, now this reduces the bulk, make the polysaccharide almost insoluble and decrease its effect on water potential or osmotic potential of the cell. They are therefore ideal for the storage and as structural component. Unlike sugar, polysaccharides are not sweet. Being large size, they are unable to pass through cell membrane, though their components do so. Passage is allowed through active process. Another method of their passage is by means of secretion vesicles of Golgi apparatus. Polysaccharides are of three main types: storage, structural, and mucopolysaccharides. You can see some differences between oligosaccharides and the polysaccharide. Now, food storage polysaccharides. They are those polysaccharides which serve as reserve food at the time of need. Storage polysaccharides are hydrolyzed sugar, thus released become unavailable or available to the living cells. Okay, storage polysaccharides, okay, sugar are thus released become available to the living cells for the production of energy and biosynthetic activity. There are two main storage polysaccharides, okay, starch and glycogen. Starch, it is the storage polysaccharide of most plants. Human being obtain it from cereals, grains, okay, for example rice, wheat, legumes, okay, that is pea, gram, beans, potato, okay, tobacco, banana, etc. It is polyglucagon homosaccharide and is formed as an end product of photosynthesis. Starch is storage either inside the chloroplast or special leucoplast called the amyloplast. Starch occurs in the form of microscopic granules called the starch granules. Starch granules may occur singly or in group. The two types are known as simple and compound starch grain. Starch grain may be rounded, oval, polygonal, or round or rod shaped in outline. Each grain has a number of shell or layer arranged in the concentric or eccentric fashion around a proteinaceous point called hilum. Starch consists of two components, amylose and amylopectin. Okay, the amylose is more soluble. Okay, it is in water than the amylopectin. Okay, then in general 20 to 30% of starch is consist of amylose, and rest is, that is about 80 to 70% is made up of amylopectin. Okay, waxy starch of some varieties of the maize and other cereal consist of entirely of amylopectin. On the other hand, starch of some varieties of pea having wrinkled surface may have as much as 98% of the amylose. So we have the various type of the polysaccharides, okay, that we are looking through. Okay, both amylose and amylopectin are found to consider, okay, condensation. They are formed by condensation of alpha D glucose pyranose form. Amylose is in the form of continuous straight but helically arranged chain, which each turn contain about 6 glucose unit. The successive glucose unit are linked together by 1 to 4 alpha linkages. That is, the link is between the carbon atom of one of and the other carbon atom 4 of the other. Okay, a molecule of water is lost. Okay, a molecule of water is lost during the formation of linkage. Okay, a straight chain of amylose consists of 200 to 1000 glucose units. Okay, now amylopectin contain a large number of glucose, you can see. Okay, glucose unit, that is about 2000 to about 2 lakh. Beside a straight chain, it bears several side chains which may be branched further. Branching of usually at the interval about 25 residues at place of origin of the side chain. The carbon atom 6 of glucose residue of the straight chain is linked to the carbon atom 1 of the first glucose unit of side chain, that is 1,6 alpha linkage. Okay, Wolfram, okay, and Thomson, these two scientists, they in 1956 also reported that 1 to 3rd linkage in case of amylopectin. Okay, test. Okay, you can see the other form also, glycogen. Okay, in this also there is branching. Fine. Test amylose fraction give blue black color with iodine solution. Okay, when we treat the amylose, then it will give the blue black color. Okay, iodine and potassium iodide solution, while amylopectin fraction will give red violet color. Okay, now glycogen, it is the polysaccharide reserve food of animal, okay, bacteria and fungi. Glycogen is popularly called the animal starch. Glycogen is mainly stored inside the liver. Okay, that is up to 0.1 kg, and muscles in shape. The complex carbohydrate appear as ellipsoid flattened granules, but that lie freely inside the cell. Polysaccharide give reddish color with the iodine. Chemically, it is similar to the starch. It has about 30,000 glucose residues and a molecular weight of about 4.8 million. Now, glucose residue are arranged in highly branched bush-like chain. They are of 2 types of linkage, that is 1,4 alpha linkage in the straight part and 1,6 linkage in the area of the branching. The straight part is helically twisted with each turn having 6 glucose unit. The distance between two branching point is 10 to 14 glucose residues. Now, inulin, it is a fructane storage polysaccharide of root and tuber of dahlia and related plant. Inulin is not mentioned or metabolized in the human blood and is readily filtered through the kidney. It is therefore used in testing the kidney function, okay, especially the glomerular filtration rate. Okay, the GFR is tested with the help of inulin. Now, structural polysaccharides. They are polysaccharides that take part in the forming structural framework of the cell wall in the plants and skeleton of the animals. Structural polysaccharides are of two main types, chitin and cellulose. The chitin, it is second most abundant organic substance. Okay, chitin is a complex carbohydrate of heteropolysaccharide. Some chitins, okay, they are considered homopolymer also, okay, which type is found in the structural component of the fungal walls and exoskeleton of the arthropods. In fungal walls, the chitin is often known as fungus cellulose. Chitin is soft and leathery, okay, leathery. Therefore, it provides both strength and elasticity. It becomes hard when impregnated with certain protein and calcium carbonate. In chitin, basic unit is not glucose but a nitrogen containing glucose derivative. Okay, and this is known as N-acetyl glucosamine. Chitin has an unbranched configuration. Monomers are joining together by 1,4 beta linkage. Adjacent residue lie at 180-degree. Okay, molecules occur in parallel and are held together by the hydrogen bond. Okay, now next we'll come to the cellulose. It is fibrous homopolysaccharide of high tensile strength which forms a structural element of cell wall in all plants, some fungi and protists. Tunicin or of tunicates, that is acidians, is related to the cellulose, also called animal cellulose. In absolute terms, cellulose is the most abundant organic substance of the biosphere, forming 50% of carbon found in the plant. Cotton fiber have about 90% of cellulose while wood contain 25 to 50% of the cellulose. The other material of the cell wall include lignin, hemicellulose, pectin, wax, etc. Cellulose molecules have unbranched and linear chain unlike the branched and helical chain of the starch and glycogen. A chain of cellulose molecule contains 6,000 or more glucose residue. Okay, then the successive glucose residue are joined together or by the 1,4 beta linkage. Consequently, alternative glucose molecules lie at 180 degree to each other. Hydroxyl group of the glucose residue therefore project in all the direction. The molecular weight of cellulose range between 0.5 to the 2.5 millions. Cellulose molecules do not occur singly. Instead of number of chain are arranged in the close antiparallel fashion. The molecules are held together by the intermolecular hydrogen bond between hydroxyl group at the position 6 of the glucose residue of one molecule and the glycosidic oxygen between two glucose residues of adjacent molecule. There is also intermolecular strengthening of the chain by formation of hydrogen bond between the hydroxyl group at the position 3 and oxygen atom of the next residue. About 2000 cellulose chain or molecules are packed together to form a microfibril that is visible under the electron microscope. Now, what is the significance? Cellulose constitute the bulk of human food. However, due to being polymer of beta glucose, cellulose is not acted upon by amylase present in human digestive juice. In human, cellulose has roughage value which keep the digestive tract in the functional fitness. Okay, cellulose is an important constituent of diet of ruminants like cows and buffalo. Stomach of ruminant contain microorganisms capable of digesting or breaking down the cellulose. Termites and snails also possess microorganisms in their gut for this purpose. Microbes are used in producing soluble sugar from the cellulose. The sugar are then allowed to undergo fermentation for obtaining ethanol, butanol, acetone, methane, etc. Cellulose rich wood is employed in the building furniture, tool, sports articles, paper, etc. Depending upon the percentage of cellulose present in the fiber, the latter are used in the textiles, cotton, linen, preparation of the sack, okay, or the rope or hemp like China jute, Deccan hemp, rayon, and cellophane are formed of cellulose xanthate. Cellulose acetate is obtained by treating the wood pulp with the acetic acid, acetic anhydride, and a catalyst. Cellulose acetate are used in preparing fiber for double knit, terracotta, wrinkle proof, and moth proof clothing. Cigarette filter are also prepared from this fiber. Other use of cellulose acetate include preparation of plastic and shutter-proof glass. The cellulose nitrate is used in propellant explosive. Carboxymethyl cellulose is used as emulsifier and smoothing the reagent of ice creams, cosmetics, and medicines. Okay, so with this we have other topic, that is the mucosubstances. Okay, mucilage, okay, mucus or slim forming substance are called the mucosubstances. They are of two types, mucopolysaccharide and mucoproteins, okay, or glycoproteins. Mucopolysaccharides are slimy substances or mucilage which possess acidic or animated polysaccharides, and these are formed from the galactose, mannose, sugar derivatives, and uronic acid. Mucopolysaccharide or mucilage are quite common in both plant and animals. So mucopolysaccharides, okay, they bind the protein in the cell wall and connective tissue. Water is held in the interstitial space due to mucopolysaccharides. Mucopolysaccharide occur in the cell wall of bacteria and blue-green algae. They form a layer of mucilage around them as well as other aquatic plants and protect the organism against rotting, okay, effect of the water, preventing desiccation also and growth of epiphytes and attack of the pathogens. They provide lubricating or lubrication in the ligaments and the tendons. As keratin sulfate, that is acetyl glucosamine plus galactose plus sulfuric acid, okay, they occur inside the skin and cornea, providing both the strength and flexibility. Chondroitin sulfate, that is glucuronic or diuronic acid plus acetyl aminogalactose is the mucopolysaccharide, okay, and it is found in the matrix of the cartilage and connective tissue for support and elasticity. Now next we have is the hyaluronic acid, okay, that is glucuronic acid plus acetyl glucosamine, okay, is the mucopolysaccharide met in the extracellular fluid of animal tissue, vitreous humor of the eye, synovial fluid, cerebrospinal fluid, etc. It also occur in the cementing material between the animal cell as well as inside the cell coat. Husk of Plantoga ovata, okay, contain mucilage, which is used medicinally in treating the intestinal problems. It relieves irritation also. Okay, mucilage prevent. Okay, mucilage, which is present in aloe barbadensis. Okay, that is aloe vera, reduce the inflammation. Okay, it also purgate. It is also a purgative. Okay, mucopolysaccharide heparin. Okay, that is alpha 1,4 glucosamine plus glucuronic acid in the blood is anticoagulant. Okay, then agar, that is murine, marine brown and red algae called the seaweeds. Okay, yield the mucopolysaccharide for the commercial value. Okay, agar, that is alginic acid, carrageenan, okay, etc. Agar is polymer of D-galactose 3,6 anhydro L-galactose having sulfate esterification after every 10th galactose unit. It is used as culture medium in the laboratory as laxative, stiffening, stabilizing, and emulsifying agent. It is obtained from the cell wall of red algae like Gracilaria, Giladium, and Giladilla. Okay. Now pectin, okay, it is an acidic polysaccharide that occur in the matrix of the cell wall and middle lamella as calcium pectide. Pectin is soluble in water and can undergo the reaction, that is salt to gel interchange. Pectin is formed of the galacturonic acid, galactose, methylated galacturonic acid, and arabinose. It is used in making jelly and jams. Hemicellulose is a mixture of polysaccharide of xylens, okay, mannans, galactans, galactans, and glucomannans. Hemicellulose occur in cell wall where it forms a link between the pectic compound and cellulose microfibrils. Peptidoglycan or the murine or mucopeptide. Okay, it is formed of heteropolysaccharide chain cross-linked by short peptide, generally tetrapeptides. Heteropolysaccharide chain are formed of two alternate amino sugar molecules, N-acetyl glucosamine, that is NAG, and N-acetyl muric, that is muramic acid, that is NAM. Lipopolysaccharide, okay, it is a complex formed of lipid and polysaccharide which forms the outer membrane of gram negative bacteria. Now, they are glycolipids responsible for the endotoxic activity, a core polysaccharide and antigen specific variable chain. Lipopolysaccharide induce fever, shock, and other toxic effects. Mucoprotein, glycoprotein, okay, the protein with conjugated mucosaccharides form mucus. These are found in the stomach, nasal secretion, intestine, vagina, and are antibacterial and protective in the function. Now, function of polysaccharides: starch and glycogen, okay, are the major storage food of the organic world. On hydrolysis, storage carbohydrate provide both energy and carbon chain. Chitin is the structural carbohydrate of fungal wall and exoskeleton of arthropods. Cellulose is the structural substance of cell wall in most of the plant. Cellulose is economically important in production of furniture, shelter, fuel, paper, textile, ropes, rayon, cellophane, okay, plastic, shatterproof glass, propellant, explosive, emulsifier, and raw material for several fermentation products. For human beings, cellulose has a roughest value, but it is a food for ruminants, snails, and termites. Mucilage present as a protective coating around aquatic plant bacteria, blue-green algae, and some animals is derived from the polysaccharides. Next is mucopolysaccharide in the human body have several functions: lubrication of the ligaments, tendons, formation of some type of body fluids like cerebrospinal fluid, and then synovial fluids, vitreous humor, providing strength and flexibility to the skin, protective flexibility to the skin, connective tissue, and cartilage binding of the protein in the cell wall and holding water in the interstitial space. Then heparin, okay, it prevents the blood coagulating or coagulation inside the blood vessels of the animal. Some mucopolysaccharide have medicinal and other commercial importance. For example, husk of Plantago ovata, mucilage of alio, and algicinic acid of agar, carrageenan of marine algae. Pectin form matrix of cell wall. As calcium pectate, they produce middle lamella or cementing layer between the adjacent plant cells. Pectins are commercial gelling agent. Now, we complete the polysaccharide with this, and we'll do the lipids in the next video.